Good morning, friends. Good morning, students. In today's class, we will discuss transcription, right? In transcription, to revise well, first, the synthesis of RNA from DNA is known as transcription, right? And from RNA, when proteins are synthesized, it is known as translation, right? And this complete is known as central dogma of human life, right? Now, in detailed terms, the synthesis of DNA from DNA is known as replication, right? And in this case, genome. The synonyms are there. The total DNA content of a cell is known as genome, right? And the study of genome is known as genomics, right? And the synthesis of DNA from DNA is known as replication, and the DNA content total is known as genome, and the synthesis of genome is known as genomics, right? And from this DNA, when we will synthesize RNA, it is known as transcription, and this RNAs formed are known as transcriptome, right? And the study of this transcriptome is known as transcriptomics, right? And from this RNA transcript will further synthesize protein, and this total protein content is known as proteome, and the study of proteome is known as proteomics, right? Or from the topic of transcription, you will get long answer questions as well as short answer questions, right? So I will start in detail now. Now I will start with some differences between replication and transcription, right? So on this side I am writing transcription. The transcription, the only a part of the DNA act as a tablet, right? It means the transcription is selective, right? And in case of replication, both the strands of DNA are involved, and they act as template. But in case of transcription, only one strand is involved, right? And for the synthesis of transcription, we are synthesizing RNA. So the enzyme is RNA polymerase, or it is also known as DNA dependent RNA polymerase. And in case of replication, you synthesize DNA, the enzyme is DNA polymerase, right? And in case of transcription, there is no need of primer. We don't need primer to synthesize RNA, but in case of replication, you need a primer, RNA primer, to synthesize the DNA. And in case of nucleotides, we have U here, right? In case of transcription, and in replication, we have T. A will combine with T by double bond, G will combine with C by triple bond, and in case of transcription, we have U instead of T, right? So that covers the major differences. Now I'll start with proper transcription. Now the main enzyme for transcription is RNA polymerase, right? And in case of prokaryotes, the enzyme is holoenzyme. And in case of eukaryotes, we have three different RNA polymerase, 1, 2, and 3. But in this class, we will discuss only RNA polymerase holoenzyme, that is in prokaryotes, right? The holoenzyme will consist of two parts. One is coenzyme, and the second is sigma factor, right? And this coenzyme contain 5 parts. One is alpha, 2 alphas are there, and second is beta and beta prime, and third is omega, right? And they have separate roles, right? The alpha will recognize the part of DNA to be transcribed for RNA synthesis, right? And this beta will help in polymerization activity and formation of phosphodiester bonds. And this beta prime will have helicase or unwinding activity that will open the strands of DNA. And the remaining sigma factor, how you can remember the function? As for sigma, as for scan, so it scans the DNA, right? And it recognizes the promoter region and helps in binding of promoter region to the RNA polymerase. So that finishes with our holoenzyme. 2 alpha units are there, 1 beta and beta prime and omega, and sigma factor that forms the complete hollow enzyme. Now to remember transcription, you have to remember this diagram first. I will give mnemonics in that also, so you can remember it very easily, right? So this is TSS. What is TSS? It is transcription start site, where transcription will start, right? So you can remember that the river Ganga starts where? It starts at Gangotri, right? So transcription, we are starting the transcription, we are starting RNA synthesis, and RNA synthesis is starting at transcription start site. So it is Gangotri for the synthesis of RNA, that is Ganga, right? So this is TSS, transcription start site, right? So Gangotri is at higher level, and when the river flows downstream, it will flow downward, right? So the site forward of the transcription start site is known as downstream, and we'll calculate with +1. We'll add the base pairs as +1, +1, +1, right? And below that, it is known as upstream, and it is denoted by -1, -1, -1, right? We'll add bases in the negative form, right? So transcription start site is that it is Gangotri, where Ganga will start, right? So transcription start site is synthesis of transcription and synthesis of RNA, right? So this is transcription start site, right? So template, I have already told, template is read in 3' to 5' direction. 3' to 5', this is our template stand, and 5' to 3', this is our coding stand, right? So this was transcription start site on -10 base pairs. -10 means we are going upstream, minus, 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 right? On -10 base pairs, we have, this is known as Pribnow box or promoter region. This base pair has sequence TA, TA, GC, and it is also known as TA, TA box, right? On -10 base pair, where the promoter region is there, and on which proper RNA polymerase binds and transcription proceeds, right? And on -35 base pairs, this red one is consensus sequence, right? Here the sequence is TT, GA, CA, right? It is -35 base pairs upstreams, right? And this is shown as red one, right? So first of all, the RNA polymerase which recognize the consensus sequence, when it recognizes the consensus sequence, it then extends its words and attach itself to the Pribnow box or promoter region, where the transcription will proceed. Now there are 3 main steps in transcription. First is initiation, second is elongation, and finally is termination, right? Now I will start with details of initiation. Students, this is TATA80, sorry, it is TATA80, not GC, right? Now the synthesis initiation, there are 4 main steps. First is formation of pre-initiation complex, then there will be formation of open complex, and then we'll synthesize small RNA nucleotide with the 5 to 10 nucleotide ribonucleotides, and then there will be promoter clearance, right? So for the synthesis of RNA, you need RNA primer, and this is you need RNA hollow enzyme, RNA polymerase hollow enzyme, right? And it has 2 parts. One is core enzyme, another is sigma factor, right? The core enzyme has 5 components: 2 alpha, 1 beta and beta prime, and omega, and sigma factor, right? Now first comes the role during formation of pre-initiation complex. There comes first the role of sigma factor, right? I already told that sigma for scanning, right? S for sigma, S for scanning, right? So this sigma factor will scan the DNA. I have written it green. Okay? It will keep on moving and it will scan the DNA and it will find consensus sequence, that is -35 base pair, and it will attach here our green one, that is our RNA polymerase, right? This is part of pre-initiation complex formation, right? So RNA polymerase is attached here at the consensus sequence, right? Then it will stretch the RNA polymerase and it will reach further at the proper, that is -10 base pair, that is TATA box or promoter region where proper RNA synthesis will start, right? Now RNA polymerase has attached first to the consensus sequence, then it stretches itself and reaches the TA box, right? And this is known as formation of closed complex. It is a closed complex, right? Now RNA polymerase is attached to the TA box where RNA synthesis will start, right? But in the second part, we have formation of open complex formation. So in the pre-initiation complex, we already formed the closed complex. Now to open it, we use helicase activity. For helicase, we'll use beta, right? The beta has helicase activity, right? So RNA polymerase will of the beta component will cause helicase activity, and there will be unwinding of the DNA strand, and this is known as formation of open complex, right? Now unwinding is there and will synthesize RNA in 5' to 3' direction. The further ribonucleotides are added to the 3' side, right? So though RNA synthesis will start and will form 5 to 10 nucleotides in 5' to 3' direction, where on 3' side further ribonucleotides are added, right? After synthesis of approximately 10 to 15 ribonucleotides, the RNA polymerase will move away from the promoter region, right? It will move away from the promoter region, and this is known as promoter clearance, right? Now we'll start with elongation. Now after initiation is second step, elongation, right? Elongation has two parts, that is polymerization and proofreading, right? In polymerization, on 3' hydroxy side, we'll add further ribonucleotides and the RNA strand will elongate, right? And in proofreading, the RNA polymerase has very less proofreading activity as compared to DNA polymerase. So the disorders are very rare, and it has not a big impact on the any disease, and that finishes with our elongation. Now we'll start with termination. The termination will consist of two parts. Now we'll start with termination. Termination is of two types. One is row dependent, another is row independent, right? In row dependent, we have a row factor, right? The row factor, this green one, attaches itself to the growing RNA chain, right? When it will reach here, it will give punch. It will give a punch to this RNA polymerase, and it will be separated from the DNA strand, and there will be termination of the transcription. So this is row dependent. Now let's start the row independent termination. Now in this row independent termination, we have formation of hairpin formation and we have palindrome sequences, right? So we are synthesizing the RNA, right? And palindromes are the sequence which you read from forward or from backward, they will remain same. For example, there is a word motor, MOTOR, and rotor, right? When you read it, you will get the same word, right? So in the DNA sequence, there are some palindromic sequences, and when they come, the RNA forms hairpin loops, right? The hairpin loop formation will give termination signals, and finally the transcription will stop, and it will lead to termination of the transcription by row independent method. So that finishes with our chapter on transcription. So please like the channel and subscribe it, because it takes a lot of effort to make such color diagrams. You've called me again and again to make videos, so I am making it. So please share with your friends and other colleagues as well. God bless you. Thank you.